Hello, everyone. I am Jacob Fries, Marketing Supervisor here at Acro Biosystems. I'd like to welcome you all to today's AcroVision webinar on advancements in antibody-drug conjugates, or ADCs, development. ADCs are a cutting-edge frontier in targeted cancer therapies, combining the precision of monoclonal antibodies with the powerful effects of cytotoxic drugs. This innovative approach allows for the targeted delivery of chemotherapy to cancer cells, reducing the collateral damage to healthy tissues, a critical advantage in cancer treatment. However, despite their immense potential, the development of ADCs remains a complex challenge. Researchers and developers face key obstacles such as optimizing the stability of the conjugates, ensuring the accurate and effective release of the drug at tumor site, and minimizing off-target effects. In this webinar, we'll be exploring the latest breakthroughs in ADC technology and the innovative strategies being employed to address these challenges. Our distinguished panel of experts will share their insights on how these advancements are helping to refine ADC therapies and bring us closer to more effective, less toxic cancer treatments. We hope today's discussion will provide you with valuable perspectives and actionable knowledge that can be applied in your own research and development efforts. I'll begin by introducing our speaker. Dr. Liu, has done postdoctoral research at LMU Hospital, Germany, with a focus on mouse lung transplantation model to discover novel therapeutic approaches for chronic lung rejection. He's also received his PhD from University of Bergen, Marie Curie ITN Fellowship in Norway, with a focus on cancer-associated fibroblast in tumor evasion and metastasis. At the end of this webinar, we'll hold a Q&A session, so please feel free to type out any questions you may have in the chat and we'll review them at the end of the session. I'd like to now hand it over to Dr. Liu to begin.
Okay, thank you, Jacob. My name is Hengshuo Liu. I'm the Technical Sales Supervisor from Acro Biosystems. Welcome joining today's webinar. This webinar will cover the following topics. I will start with a background overview of the ADCs and then discuss the challenges and solutions for ADC development. So first, let's take a look at the market share of ADCs. The figure to the left shows all the approved ADCs and those from phase 3. In 2023, it has reached almost 9 billion, 10 billion US dollar. And by 2028, it is predicted to reach $26 billion of market revenue. And ADC has the fastest growth rate in pharmaceuticals. If you look at the picture to the right, here lists the pipelines that is under development. We can see that the majority of the ADCs from phase 1 to phase 3 pipelines target different solid tumors. Breast cancer, lung cancer, gastrointestinal cancer, they all have the highest pipelines. Furthermore, you can also find pipelines for ovarian cancer, pancreatic cancer, etc. This indicates that ADCs are being positioned as an essential tool in the precision oncology therapeutics. For clinical treatment, chemotherapy is one of frequently used approach. It is designed with molecules that can disrupt cell cycle and cell proliferations, but they don't have target specificity. And because of that, there's always a need to balance the toxicity and dosage, therefore limits the efficacy. Monoclonal antibodies has also been used for clinical treatment of cancer. They have high specificity towards target antigens, and they are designed to stimulate, inhibit, or block signal along the signaling pathway. They also face challenge of clinical relapse due to tumor turnover. ADC is a combination of monoclonal antibodies and chemotherapy. It is designed to deliver precision cytotoxic payload with minimal off-target and maximal on-target effect, aiming to broaden the therapeutic index. So we have known that ADC has a very large market share. It has very fast growth rate and many pipeline under development. What is the reality of the development? It looks discouraging that up to now only 15 target has made to the approval, and 92 pipeline has been discontinued, and there are in total 267 from the pipeline. The percentage of ADC approval is low. This is due to the challenges that people are facing during the development, including insufficient therapeutic benefit due to toxicity, or the therapeutic benefit from ADC doesn't show superiority to conventional therapies. The DAR heterogeneity causing inconsistent cytotoxic effect. The ADC is rapid clearance from the body due to the poor PK and off-target toxicity. So why we are facing so many challenges? This is because there are so many different components in the ADC. For example, the antibody affinity, specificity, the conjugation choice, either is non-specific or specific conjugation. The linker of choice is cleavable or non-cleavable, and we have many different choice of payloads. Therefore, to overcome these challenges, we are offering different solutions. I will go through them one by one. The first challenge is managing the target affinity. So antibody affinity is not only crucial for mitigating on-target and off-target cytotoxicities, but also crucial for ADC effectiveness. Affinity strategy for ADC development is different from conventional antibody development, where highest affinity is desired. For ADC, if the affinity is too low, it can result in insufficient binding or internalization, especially when the antigen expression is low on the target cell. Conversely, if the affinity is too high, it can lead to reduced receptor occupancy and internalization. Affinity validation is mostly done with SPR and BLI. So here is a case study. When you have a large library of compounds, antibody, or ligand, then high throughput SPR screening is a very fast solution providing a fast screening. This is a collaboration with Katara of high throughput screening of antibody candidates against full-length CCR5. The target proteins provided by us. You can see that you can, with high throughput SPR, you can fast generate of hundreds or thousands readouts from the candidates. Besides of the antibody and target affinity, the FCR affinity is also crucial for antibody optimization. When the IgG binds to FcRn, its half-life in serum is prolonged, and therefore the PK is also prolonged. When binding to FcR gamma, it leads to ADCC and ADCP, which are also important for antibody characterization and validation. So here is another case study of a high throughput screening of different FCR product provided by us. And you can evaluate the binding affinity and select the best candidate from that. SPR can provide us with antibody affinity at molecular level. To further analyze the binding at cellular level, the selected candidate from high throughput SPR proceeds to ADCC and ADCP evaluation using reporter cell line. So here I'm showing two data, and using our reporter cell line. The figure to the left is ADCC activity evaluation using human CD168 JERCAT reporter cell line. And the figure to the right is a ADCP activation evaluation using human CD32A JERCAT reporter cell line. So this data can show you the binding affinity at cellular level, which is crucial for clinical trial. The next challenge is DAR inconsistency and conjugating different payloads. Conventionally, the conjugation targets lysine residue and reduced interchain disulfide bonds. This leads to different DAR and linker precision, causing inconsistent DAR and efficacy. Therefore, site-specific conjugation technologies have been used to produce a more homogeneous ADCs, and it has been shown that the PK is improved and it can provide better therapeutic index. When it comes to payload, there are many different types to choose based on their function. So you have tubulin inhibitors, DNA damaging agent, immunomodulators, topoisomerase inhibitors, and there are many more under development. When it comes to conjugation, therefore, we need a technique that can accommodate a broad range of payload choice. And this is the kit, the conjugation kit we offer that can achieve DR2 and DR4. And this kit is capable of conjugating MMAE or biotin payload or conjugating other different payload via pyrazine or DBCL functional group and click chemistry. So this is some case study, and these are the DAR conjugate with our kits. We have validated first the homogeneity of the conjugation, so DR2 and DR4. We also validated the stability in plasma. We can see that from day 0 to day 8, there's no dissociation of the payload from the conjugation. And from the aggregation analysis, we can see that there's very minimal ADC aggregation from the conjugation. Furthermore, we also validated the antibody bioactivity. First figure shows that binding to conjugating to MMAE doesn't affect the binding specificity. So this is the trastuzumab conjugation. We also validated in vitro cytotoxicity, showing that the conjugation can deliver precise cytotoxin to the target cells. So there's no effect to HER2 negative cells. Okay, we move on. The next two challenges are linker efficacy and in vivo PK evaluation. So for linkers for ADC includes non-cleavable linkers and cleavable linkers. Cleavable linkers are designed to be able to be stable in blood serum and release the payload inside the cell. Dipeptidic linkers are commonly used as a cleavable linker that targets cathepsin B, and cathepsin B is upregulated in many different cancer cells. So if the ADC is linked with cathepsin B target linker, once the complex is taken by the cells, so the payload can be released from the lysosome. And linker cleavage evaluation, it is very important to validate payload delivery. This is another case study. The client has a HER2 antibody conjugated to DXd with CK linkers, which is cleavable by cathepsin B. And we provided the cathepsin B for the evaluation. The figure to the right are the data from CMS provided by CMP Scientific. The top one is from the control group showing the intact ADC. And the figure to the bottom is the ADC after cathepsin B digestion. We see there's a additional peak to the left. That is the antibody released from the payload. and the payload peak is further to the left side. So this figure shows that our cathepsin B can release the payload from the ADC conjugate, and showing that the payload can be released inside the cells. About evaluating the in vivo PK of ADC, it is more complicated comparing to conventional drugs because there are different components, including like antibody and payload, etc. Therefore, the PK for ADC resulting in diverse drug delivery profile over the time. The PK assay of ADC generally are configured to measure all forms of ADCs, total antibodies, and release payload. Based on the targets, there are two types of ADC PK evaluation. First one is the PK evaluation targeting antibody, including total antibody and conjugated antibody. And they are analyzed by ligand binding assay. The second PK is targeting payload, including free payload and conjugated payload that are analyzed by LC-MS. The figure to the bottom showing the different ligand binding assay format. So based on the readout, you can have like anti-idiotypic antibody captured on the plate or antigen target on the plate, or it's anti-payload antibody captured on the plate. So by this different combination can generate different PK results. This is a 2 case study validating the PK. The figure to the left is has immobilized mouse payload antibody, which is anti-DXd. Then the ADC is added to the system, then a detecting antibody is added. The figure to the right is a different design where the antigen is immobilized and binding to the ADC, and on top of that, the detecting antibody. So these are 2 examples of different LBA formats. Okay, from Acro, you can find ADC solutions for different development stages. For early stage discovery and development, we provide you with ADC targets, the conjugation kit, proteases, for example, cathepsin B, MMPs, Fc receptors, overexpression cell line, SPR-BLI analytical service. For manufacturing and QC, we provide you with nuclease, residual whole cell DNA detection kits, mycoplasma detection kits, and quality characterization analytical service. For preclinical studies and clinical studies, we offer you anti-payload antibodies, anti-idiotypic antibodies, PK ELISA assays, cytokine release assay kits, which is ELISA-based. We also have a multiplex cytokine assay kits. We can also customize anti-idiotypic antibody. In addition to ADC development, we also offer a broad collection of products and services for antibody-drug and cell gene therapy development. The reason to choose Acro Biosystem is that we provide a wide collection of products and custom solutions from RUO to GMP grade to support you from early stage discovery to clinical trial. Our products and services have high flexibility. You can choose expression system between hex cell, CHO cell, E. coli, insect, and you can choose like the tag for the protein. And you can also require, for example, specific QC to validate the performance of the product. And we also offer a fast communication. We have dedicated project manager and we have the sales team globally. And we can also offer scale-up production based on your needs. So for GMP production, we offer up to gram level. Okay, and our principle is that we our product and service always have better design and better quality, and we provide you better support and better response. Okay, that's the end of the webinar. If you want to learn more about our product and services, please feel free to scan the QR code and contact us via email. Thank you for listening. I'm happy to answer your questions.
Thank you so much, Dr. Liu. We'll now open the Q&A session. So please, if you have any questions, feel free to drop them in the chat and we'll go over them. So we do have one question. Can the ag-link conjugation kit be used for PROSEB scale-up production?
Yes, it can. It can be used for a scale-up production up to milligram or gram levels.
And what type of payload antibodies are currently available?
We have several anti-payload antibody off the shelf, including MMAE, MMAF, DXD, SN38, etc. And we also offer customized anti-payload antibody service.
Can you elaborate a little bit more about one step, one pot, and how to achieve that of the ag-link conjugation kit?
Yes. So this kit includes several enzyme and substrate needed for the conjugation in a one mixture. And this compound work together. That's why it says one pot. And then it can, so this compound work together in a method to modify the antibody's Fc domain, the N-glycan, and attach the payload of your choice.
Great. Are these conjugation kits suitable for antibodies from all species?
Yes. This conjugation kit is highly effective for humanized antibody conjugation, and it has been validated. For other species, for example, marine rabbits, you might need to extend the conjugation time because their complex structure and incomplete validation. But the answer is yes, it is suitable for different species.
Great, thank you. Can this kit generate dual drug ADCs?
Yes, for dual ADC conjugation, you can use the DBCO or TZ conjugation kit to connect specific linkers that are already attached to different payloads through click chemistry.
And then which of the process development using different site-specific conjugation methods is easier to scale up?
So each site-specific conjugation has its pros and cons. Take enzyme like enzyme cleavable conjugation, for example. It is scalable. During the production process, you need an extra purification to remove the residue enzyme.
Perfect. We have a few more coming in. One moment. I do see one question on how to get access to the slides. So we will send those out after the webinar.
I think maybe there's an important message deliveries here. here is that we do not offer ADCs. We provide different raw material that is used for ADC research and development.
I have another question. Oh, no, disappeared. Okay, so what about analytical challenges and characterization approaches?
So for antibody, let's say specificity validation, so the commonly used techniques SPR and BLI. And once you have the candidate, then you continue to the cellular level using like reporter cell line to validate the binding to, for example, FCR gamma to confirm the ADCC and ADCP. And for example, the PK analysis challenge is that you have to find the correct setup. For example, there are so many different ligand binding assay. So depending on, let's say, either it's targeting the payload or targeting the idiotypic region, or it's like coated with antigens, they generate different readout and they tell you the PK from different aspects. So we can provide you the solution based on your needs. So please feel free to contact us so we can design assay for you.
So we have another question, whether you provide support to academic institutions.
For academic clients, we always offer a discount to support the innovation there. And then, so in this case, please feel free to contact our sales team or contact me, then contact us, then we will direct you to the corresponding sales team. Then they can, based on your needs, so we can provide you with tailored service.
Great, thank you. We have another one. Do you think ADC can be bound with cell therapies, NK or T cells to kill cancer cells?
Oh, that's a very interesting question. So that means it's a combined therapy, right? I think it is possible because cell therapy has shown promising effect on blood tumor like lymphoma, etc. Well, from the pipeline, we see that ADC has more advantage on solid tumor. And but clinical application, it's very difficult, I'll say, subject based on the doctors like practice preference. Also, we also need to, how say, offer more training, introduction to tell them the advantage of each approach. But I think this question we need more discussion with like more experts. But it's a very good question. Thank you.
Do we have any further questions?
So I see there's a question, do antibody work on intra or intracell surface like molecular mortar? So I guess you mean that if the antibody will intake by the cells? The answer is yes, because for ADC, treatment is important to validate the internalization, because that's when the whole complex enter the cell, and then it goes into the lysosome, and the cathepsin B will release the payload from the complex, and the payload can further be delivered to, let's say, nuclear or to the cytoplasma to reach their target. There's a question if we quality control by native mass spectrometry as well. We don't have an MS technique, therefore we cannot validate our product with MS. There's one question about, I think the main limitation of ADC is after injection to the patient, many ADC will focus on one cancer cell, but other cancer cell will be free. How to overcome this problem in clinic? This is also a very important question, because ADC is antibody-mediated delivery of payloads. So I think it starts from the antigen screening, that you want to select, let's say, the antigen that is abundantly expressed on the cancer cell and also very low expression on the tumor, on a normal tissue. And on other type of cancer, then it's about that if this antigen express, let's say, different stage of the tumor, or let's say multiple different tumor. But from my knowledge, I don't have like a clear overview that for each, let's say, approved ADC, how many different tumor they can be treated. But this also comes with that the discussion of combined therapy. That, for example, do we use like more than one ADC for the treatment, etc. But that also comes to the further question that what the balancing the cytotoxicity side effects from the patient. Yeah, so for that, I don't have a clear answer for that, but this is an important question for clinical practice. There's a
Another question is, what is the role that the linker will play? Can we attach the antibody directly to the cytotoxic payload?
So the conjugation occurs on the side chain of the Fc domain. And the linker, the reason to include linker is that we need to release the payload from the ADC. That's why, for example, we can choose a enzymatic linker that can be cleaved by cathepsin B, and cathepsin B is upregulated in many cancer cells. If the payload is directly, say, linked to the antibody and cannot be released, then ADC cannot, say, induce cytotoxicity, and then the tumor cell will not be able to be eliminated.
Great, thank you. Do we have any other further questions? I see typing. Okay, so if we have no further questions, then I think we'll end here. I'd like to thank you all again for joining us today. You'll get a copy of the slides after the webinar, and please feel free to reach out to Dr. Liu for any further questions.
Okay, thank you for joining. Hope you have a nice day.
Thank you all very much.